Hello, everyone, and a warm welcome to our webinar today. I am Bruna Lousada, the European Manager at Hansen, the Teledyne Labs division specialized in dissolution and diffusion test systems. I'm thrilled to introduce today's session, Advances in In Vitro Bioequivalence Assessment for Topical Products, with IVRT focus with Dr. Theo Capanazzi, co-founder and Chief Scientific Officer of DITIBA. This will be the first part of a two-part series. The next session will focus on IVPT, and we'll be sending out invitations in the coming weeks. Some of you might remember Theo from our very first diffusion webinar in 2020, which also focused on in vitro bioequivalence testing, but for ophthalmic products. If you're interested, you can still watch it on the Teledyne Hansen YouTube channel, but today we will dive deeper into in vitro bioequivalence and cover everything you need to know about conducting a successful IVRT study. For those who are not familiar with Theo, he is the co-founder and chief scientific officer of DITIBA and has played a key role in establishing the company as a leading CRO in dermal and transdermal research services. Under his guidance, DITIBA has established a strong track record in characterizing topically applied products and possesses superior scientific expertise in vitro release tests, in vitro permeation tests, and the methodologies developed at DITIBA have contributed to numerous innovator and generic drug approvals. So before we begin, a few housekeeping notes. All attendees are currently muted, but you can submit your questions at any time using the question function on your screen. We will address as many questions as possible at the end of the presentation, and if we run out of time, we will follow up via email. If you experience any issues with your audio or connection, please try logging off and logging back on. For those who can't watch the entire webinar or wish to revisit later, a replay will be available on the Teledyne Labs YouTube channel in the coming days. With that, I'm delighted to hand over to Dr. Theo Capanazzi.
Yeah, thank you, Bruno, very much for such a nice introduction. Hi, everyone. I'm thrilled to be here today to talk about specific regulatory approval for topical products applied to the skin. As Bruno mentioned, I'm Theo Capanazzi from DITIBA Analytical Service CRO based in Toronto, Canada. It is a pleasure to discuss the role of in vitro IVRT and IVPT methods as a surrogate alternatives in the development and approval of generic topical products. As we all know, pharmaceutical companies face stringent regulations, high cost, and significant time investment to bring their drug products to the market, allocating substantial resources to demonstrate the quality, efficacy, and safety. And any products is essential before regulatory agencies grant marketing authorization. Therefore, understanding the specific requirements and consideration of each regulatory bodies is crucial. As many of you know, after patent and exclusivity expire, company can produce generic drugs. When two drugs are proven to be bioequivalent, regulatory bodies consider them therapeutically equivalent and allow the new formulations to be sold based on the original safety and efficacy data. Bioequivalence essentially demonstrates the similarity between products being compared. There are two primary ways to conduct bioequivalence trials: in vitro and in vivo. Regulatory agencies such as US FDA and EMA in European Union allow both in vitro and in vivo bioequivalence approaches. Depending on the nature of topical products, complexity of the formulation, and targeted sites, is it dermal, local application, or is it transdermal? either in vivo and in vitro approaches could be applied for B studies. These B options are often specified in the product-specific guidance, so-called PSGs, provided by the FDA for topical drug products submitted in NDAs. Generic drugs play a vital role in understanding healthcare costs, challenges, and increasingly accessible essential medication to the patients worldwide. However, manufacturing dermatological products is complex due to intricate microstructure. It's very complex, varied excipients, and diverse delivery mechanisms. It is cream, gels, ointments, etc. Specific manufacturing equipment is required, which can lead to monopolistic behavior by innovators, resulting in the high cost for patients. It is a fact that large percentage of dermatological drugs do not have generic equivalents on the market in the United States and in Europe. Assessing bioequivalency of locally acting topical products is challenging due to their low on undetectable systemic drug levels. Traditional in vivo B studies using pharmacokinetics, PK endpoints, Cmax, and AUC are often not feasible for these products. For transdermal products, PK data can be used, but dermatological skin drug levels, dermatokinetics are desired, which is challenging to measure. In US FDA 21 CFR 320-24 outlines guidance for bioequivalent topical products. Each B options has benefit, risk, and limitations, and not all are suitable to every generics. Historically, clinical endpoint studies were the default for evaluating B of topical products. However, these have issues. For example, disease skin can alter barrier properties and permeability, high placebo effects, subjective assessment, a patient's compliance issue, require a large number of the subjects, it's time consuming, ethical, economical, it is expensive too. The FDA considers clinical endpoint B studies as the least favorable in vivo options. Prominent alternative B methods include IVRT and IVPT. Other recommended options are, as listed, tape stripping, dermatopharmacokinetics, microdialysis, microperfusion, Raman spectroscopy, near-infrared spectroscopy, physiologically based pharmacokinetics, and the in vitro in vivo correlation. Each methodology, the applicability for B assessment is discussed extensively in literature, included work by FDA research team. Now, influence of excipients on bioavailability. Besides the nature of active substance APIs, excipients, inactive ingredients could influence on bioavailability, efficacy, and safety of locally acting products. Excipients could enhance stability, solubility, bioavailability, and overall product's performance. In skin applied products, they play a crucial role in drug release, skin permeation, and site-specific effects. Now, role IVRT, IVPT method is a surrogate alternatives. Normally, IVRT examines drug substance release from the topical formulation over time, offering insights into the release kinetics. Its use synthetic membranes instead of skin, and mainly useful for quality control, detecting formulation changes, lot consistency, post-approval adjustment, and at the time, bioequivalence evaluation. When IVPT assess drug permeation through the skin layers, mimic absorption and distribution into the skin. It employs human skin membranes and predict product's behavior upon application. In terms of bioequivalence, IVRT compares release profile to ensure consistent drug release, indicating similar efficacy and safety. IVPT demonstrated comparable permeation rates and extent to achieve drug absorption through the skin. Thus, IVPT evaluated drugs availability at the skin site of actions, characterizing the comparable bioavailability between test and reference products. We do have regulatory guidance, which is shown on this slide. EMA draft guidance on topical products quality and equivalency since 2018, two FDA guidance, IVRT and IVPT separately, effective since October 2022, and recently revised USP Chapter 7024 includes IVPT for the first time, offering in vitro performance tests for semi-solids and liquid-based dosage forms. Importantly, both FDA and EMA provide standards for evaluating and using in vitro testing for bioequivalence in NDA submissions. Benefits in vitro studies is this is cost-effective, time-saving alternatives to the clinical studies, offering controlled and reproducible bioequivalent assessment, accelerates products approval, reduce research cost, and speed up bioavailability and the generic topical products. Regardless, regulatory agencies mainly harmonize IVRT and IVPT methodologies. There are also differences. The major differences in agency approaches is that FDA gives detailed product-specific guidance, while EMA adopts a broader perspectives. Choice relevant in vitro diffusion systems. Selecting the appropriate diffusion systems is crucial in the development and in vitro release method. Various in vitro release testing systems are proposed by industry focusing on reproducibility and discriminatory capabilities. Common IVRT systems are presented on this slide and well defined in mentioned USP General Chapter 7024 and FDA guidances. First is a static French diffusion cells, traditionally a preferable choice, especially for dermatological products, providing reliable results and has history of successful IVRT method development. Second is a USP Aperture 2 immersion cells and Aperture 4 semi-solid adapters are used for a range of the products. Aperture 4 may be suitable for ophthalmic or buccal sublingual products where most of the API is absorbed systematically. Automated testing systems, full automated systems like Phoenix from Teledyne, alternatively we have Logan or Permigate, offers advanced capabilities. Automation provides advanced over the manual methods in terms of technical handling, compliance, data management. Validated software allows for secure storage and testing conditions, or trail for regulatory purpose and easy method transfer, ensuring standardized condition in operation protocols make it easy to reproduce results consistently. Automated system streamline IVRT experiments, enhance data accuracy, ensure compliance, and facilitate regulatory approval by providing robust data management capabilities and reproducible testing conditions. Selection and optimizing IVRT condition. First, of course, is a receptor medium. Choice receptor medium based on drug solubility and stability. Must ensure stability at sink conditions. Ideal receptor medium are aqueous buffer solution, PBS for instance, pH 7.4 buffer. However, for non-aqueous medium may be needed for water insoluble drugs. adding surfactants and using solvents like alcohols and propylene glycols can facilitate drug release. Second is a membrane. Evaluated synthetic membranes of different size for inertness to both receptor medium and the formulation. Examples are nitrocellulose, Durafor, Topfrin, nylon, and etc. We have lots of other alternatives. Second is, I will consider also matrix specifics. Consider matrix properties. For example, rheological behavior when developing IVRT methods is crucial. Fast release matrix can require early sampling time points, for example, from 0 to 2 hours for better discrimination. Slow release matrix need carefully considered to avoid prolonged experiment that's not mimic in vivo conditions. Typically, IVRT duration according to SP 7024 is 4 to 6 hours. EMA suggests a duration sufficient to achieve more than 70% dermatological product released. At least we need 5 time points are needed for a linear regression part of the IVRT slope. Technically, Higuchi's model applies a pseudo infinite dose model, require a high initial access of the drug. Steady state release kinetics are usually assumed with dose depletion less than 30%. Dose application variability has to be considered. EMA requires cell-to-cell variability within plus minus 5%. This resolved with accurate dosage applicators. Relationship between dose amounts, release rate, and dose depletion is crucial that has to be considered. Once the IVRT method is developed and validated, considering the optimal conditions, it is essential to understand both FDA and EMA guidelines. For method validation, it's important. There are some shared and divergent requirements, critical for these studies, NDA submissions for and marketing authorization. Let's see the validation parameters. First is the linearity and range. FDA's linearity R square more 0.97 evaluated over the 6 or 8 hours. EMA's linearity R square is more liberal, 0.9, with importance of statistical significant slope. Second is a precision and reproducibility calculated for each diffusion cells. Mean standard deviation and percentage CV are measured. For FDA inter and intra run, CV should be less than 15%. When EMA's intra and intra run, variability should be less than 10%. Included intermediate procedure studies with different operators and the days, CV less than 10%. Dose depletion characterized by the recovery of the drug in the receptor solution over the IVRT duration. FDA typically dose duration less than 30%. If more than 30%, it is okay, but evidence must be show a suitable linear release rate slope. EMA, as mentioned, require more than 70% of the applied drug release, often not feasible for dermatological products. It's a second issue, it's a discrimination, which is conducted through the sensitivity, specificity, and selectivity. FDA requires demonstration of sensitivity, ability to detect changes in release rate, and specificity, linear response to API amount changed, based on the at least one run of the 6 diffusion cells for each 3 product strengths, 50% labor claim, 100% labor claim, and 150 labor claim. EMI requires also 3 strengths altered formulation: 50% labor claim, 100% labor claim, and this time 200% labor claim. And requirement linearity is more liberal. This is a more R square should be more than 0.9. EMA is asking for demonstration ability to discriminate changes in Q2 composition, manufacturing process, particle size, and rheology. FDA specified as a supplementary selectivity. The standardized IVRT method validation parameter listed on this slide. Again, ensure low intra and inter-batch variability. Accurate dosage to maintain cells-to-cells variability within plus minus 5%. Managing differences in required dose depletion and release rate linearity criteria between the FDA and EMA. Detailed justification for each selected IVRT duration, dose depletion criteria, and discriminative power must be provided to meet both FDA and EMA requirements. The linear correlation for IVRT validation works exceptionally well for many dermatological products, as displayed on this slide, including those products outlining in product-specific guidance. We have successfully filed numerous NDAs for various products with IVRT method, encountering no major quality deficiencies. However, there are cases where challenges arise require creative approaches to overcome the problem. I want to highlight some of those cases and propose potential solutions. This is case 1 study. Challenges with saturated API level in some formulations. The active pharmaceutical ingredient API is presented at saturation levels, either dissolved or suspended. Additional API may be precipitated out, compromising the matrix. We encountered difficulties once in preparing even 120% labor clay products without destabilizing the matrix. Microscopic examination revealed clumps and large API aggregates, which result in inconsistent release rates and high cell-to-cell variability. Thus, in such cases, the key issue are high variability and inconsistent slope. Difficulties to demonstrate discriminatory power between 100% labor claim and 150% labor claim. Potential solutions are the first, prepare series altered loads. For example, prepare 50% labor claim, 75% labor claim, 100% labor claim, and 125% labor claim formulation, if you may. Demonstrate trends in increased rates as a function API concentration. Aim for acceptable linearity to show discriminative power of the IVRT methods in response to API amount changes. Second, might be considered a narrow confidence interval. EMA proposed, as we know, 90% to 110% instead of broader 75 to 133% confidence interval. We are lot with 125% failing to comply. However, this should be cautioned. More restrictive confidence in a plus minus 10 acceptance range may be compromise equivalence demonstration during the pivotal IVRT study. Therefore, still consider using statistical methods like the Wilson rank sum mean witness rank test proposed by FDA. I believe it's a best choice there. This is case study 2. Challenges with slow and controlled release matrices. In products, matrix with slow and well-controlled release characteristics, increasing the API concentration may not necessarily yield a proportional increases in the release rate. For example, doubling the concentration from 100% labor claim to 200% labor claim often does not result in doubling the slope. Instead, increases follow the square root relationship. We are doubling the API concentration, only increase the slope by approximately 1.4 times, square root from the 2. Consequently, the Wilson rank sum Mann-Whitney's rank test at 90% confidence interval allow an altered load with 150% labor rate to fall within 75 and 133, which is deemed too permissive by FDA USP standards. For such formulation, only load with more than 200% labor rate typically falls outside the acceptable range 75 to 133. This is nonlinear response to product strengths does not necessarily indicate lack of sensitivity in the IVRT method, but rather reflects the intrinsic slow and controlled diffusion process of the matrix. Well known full dissolved API in the matrix, the slope release rate is proportional to the amount of the API. But however, partially or fully suspended API, the diffusion kinetics are more complex, following the square root relationship instead of direct proportionality. Even with optimized IVRT method, statistical comparison slopes ratio for altered load 150% versus 100 is main cases still falls within the broad range, 75% to 130 range. Mediation strategies. First is again prepare series of altered loads with varying API concentration. This is 50%, 75%, 100%, 125, 150%, 175, 200, perhaps 225 labor claim. Demonstrate a clear trend where the low API concentration correlates with the low release rates and high concentration yields higher slopes. Ensure variability exceeds the method's reproducibility, inter and intraday precision, and falls outside error bars. Here is an example displayed on this slide. Only lot with 225% labor claim and above falls outside the specified range 75 to 133%, satisfying the FDA requirements for method discriminatory power. Second options I would consider is alternative statistical approaches. Consider applying additional statistical techniques to demonstrate sufficient differences among the lot. Maybe increase cells count. Use more than 6 cells, for example, 12 or more, for robust comparison data to enhance method sensitivity. By implementing these strategies, the IVRT method can better exhibit its discriminatory power and align with the regulatory expectation and ensure reliable performance across various products' strengths. As this is the last case study I would like to present is viscosity builder APIs. This is very rare instance. The API itself acts as a viscosity builder in the formulation. Hence, changes in amount of API affects the product's rheology significantly. For lots with 50% labor claim and the 150% labor claim, viscosity can be vary greatly from the reference product, which is 100% labor claim. This leads to the inadequate response rate and API concentration changes. Challenges is adjusting viscosity while maintaining Q1 and Q2 equivalence. Plausible solution is a two. Introduce additional viscosity adjustment. Add viscosity adjustment ingredients do not present in RLD in formulation with 50% labor claim and 150% labor claim. That requires regulatory approval for sure. Second is a variation in active ingredients. Since API adjustment are impractical, vary amount of inactive ingredients. Method should be respond well to changes in active ingredients, demonstrating supplementary selectivity. Thus, summary and conclusion, it's determination of the IVRT release profiles is crucial throughout the product lifestyle. During product development, it provides essential data on the product's microstructure, aiding optimization of both formulation and production process. In the later stages, development and post-marketing phase, it serves as a quality control tool to ensure batch-to-batch consistency. Advantages of IVRT methods is high sensitivity and discriminatory power. Thus, IVRT has three major applications. This is a product development, helps optimizing formulation and manufacturing process, quality control, ensure consistent products quality and performance over the different batches, and bioequivalence demonstration used to compare new products with the pre-approved ones where applicable. Regulatory expectation. Increasingly, regulatory bodies mandate IVRT as a quality tool, expected to be part of semi-solid QC release and stability specification. Traditionally performed using vertical diffusion cells, French cells. Now moving toward the automation for improving throughputs and reproducibility. Thank you, and back to you, Bruna.
Thank you so much, Theo, for this excellent presentation. We will now move on onto the Q&A session, and I would like to remind everyone that you can submit your questions using the question function on your screen. So we already have a few questions submitted. First question for you, Theo. What are the key differences between in vitro and in vivo bioequivalence studies for topical products?
Thank you for addressing such complex question. Yeah. When considered bioequivalence studies for topical products, multiple factors needs to be taken in consideration. The primary objectives of in vitro approaches is to evaluate the release and permeation characteristics of the drug from the topical products without involving human subjects. This approach typically requires less time and resources compared to in vivo studies. As mentioned in my presentation, in vitro study involves the use of laboratory assays such as IVRT and IVPT, which typically utilize synthetic membranes or biological membranes. In contrast, in vivo study involves clinical trials with human participants to evaluate the product's performance on the skin. In vitro data can guide formulation development and optimization, helping to identify formulations issue before conducting in vivo studies. In vitro tests are generally more straightforward, focus on the physicochemical properties. On the other hand, in vivo studies aim to demonstrate therapeutical equivalency of the topical products in humans by assessing safety and efficacy, which provides clinical relevance. In vivo studies are usually mandatory for final regulatory submissions for bioequivalence, especially for complex formulation where systemic exposure is significant.
Thank you, Theo. Next question here. Are there any recent updates or changes in the guidelines from the FDA or the EMA that we should be aware of?
Yeah, certainly. There are some important updates to note. The FDA recently updated its guidance in October 2022, separated these guidances for IVRT and for IVPT, for topical products submitted abbreviated new drugs application, NDAs. Additionally, the United States Pharmacopoeia, USP, updated General Chapter 7024 in current years, which covers both IVRT and IVPT procedures. These updates emphasize the applicability of the product performance test as well as a bioequivalence assessment. Regarding the European Medical Agencies, EMA, there have been guidelines in place since 2018 concerning the quality equivalency topical products. These guidelines remain crucial resources for ensure compliance with the EMA standards for topical drug products.
Thank you, Theo. Next question we have here. How does IVRT correlate with in vivo bioavailability or bioequivalence?
Yeah, normally IVRT is primarily quality control tools that ensures batch consistency, evaluates the release characteristics of the drug from the formulation. While it does not directly predict in vivo bioavailability, it sometimes can be used as a surrogate to support bioequivalency. Examples is, for example, acyclovir ointment. Yeah. IVRT is particularly important for demonstrating that the generic formulation is equivalent to the reference product in terms of the drug release. This is often part of evidence required for NDA submissions for a filing. Yeah, IVRT is mostly suitable for semi-solid formulations such as creams, gels, ointments. It may be not effective for a formulation like transdermal patches or liquid solutions without appropriate modification to the testing unit. That's all.
Thank you. Next question. What are the main challenges in selecting an appropriate membrane for IVRT, and what other factors impact the reproducibility of IVRT results?
Yeah. Challenges in selecting appropriate membranes for IVRT. Yeah, first of all, first is a membrane compatibility. Membrane must be compatible with both drug products and receptor solution to ensure accurate testing. This includes avoiding chemical interaction that could affect drug release. Second, I would consider a drug binding avoidance. It is critical to select a membrane that does not bind the drug, API, as it can alter the release profile and lead to inconsistent results. Third one I would like to mention is a resistance to API release. The membrane must provide low and consistent resistance to the drug release to ensure reliable measurement. Porosity as thickness. Consistency membrane porosity thickness is essential. Variation can significantly affect drug release rates and overall reproducibility of the result. Changes, let's say, suppliers or discontinuation of certain membranes can impact of release rate. For example, it's possible to experience differences in IVRT results due to changes membrane ingredients, such as was case for one lot with a nylon membrane, showed twice differences in slope compared to previous results. This highlights the importance of suppliers' transparency and quality control. From the other factors impacted IVRT reproducibility, I will highlight dose application techniques. The method of applying the dose can affect consistency of the result. For example, squeezing products from the commercial tubes or syringes could disturb formulation rheology when you apply, leading to variability. A consistent application techniques validated during IVRT method validation should be maintained throughout the testing process. Other factors I will mention is the temperature, receptor solutions composition. Both can significantly impact drug release rate. This is critical to maintain precision temperature control and use consistent receptor solution compositions throughout the test. Other factors, the mixing rate. Constant and homogeneous mixing is necessary to ensure uniform distribution of the drug in the receptor solution and prevent localized concentration gradients that could alter release rates. Diffusion cells dimension also important to maintain diffusion dimensions. Because variation leads to differences in surface area and consequently affect to release rates. Last one I want to mention is environmental control. Consistent environmental control such as humidity and temperature necessary to prevent external factors from influencing the results. I hope I did not miss any important factors.
Many factors can impact. Next question we have here, Theo. What resources or infrastructure are essential for conducting a reliable IVRT?
Resources. Conducting reliable in vitro requires specific resources infrastructure to ensure accuracy and consistency. Key elements are this is a high quality diffusion cells, of course. Reliable diffusion cells with consistent geometry are crucial for accurate measurement. These cells should be maintained properly to ensure their dimensions and integrity remaining unchanged. Precision instrumentation, every lab has this is balances and other lab equipment such as chromatography systems and etc. Important is automatic systems. Automated systems and mixing systems can improve consistency and reduce variability due to manual handling. Standard operation procedure, SOPs, and validated protocols should be in place for any labs conducting IVRT, including sample preparation, dose application, membrane selection, analytical methods, etc. Qualified personnel, skilled personnel trained in IVRT methodology are critical to setting experiments, operating equipment, and analyzing the data correctly. It is necessary to have data management system needed to accurate data collection, storage, analysis, and ensure traceability with the regulatory standards.
Those are the key things we need to have.
Thanks,
Theo. And then here I have another question. So how do companies typically address regulatory feedback on IVRT data submission?
To effectively address, I believe, regulatory feedbacks, IVRT submissions, companies often follow the key practice here. First is understanding regulatory expectation. Companies should stay informed about current regulatory standards and guidance. Recently, FDA has standardized the submission requirements for topical products modules for IVRT and IVPT. Participation FDA workshop is valuable for understanding what to submit and where in application to place this data. And documentation, it's important. As common reasons for IVRT data submission deficiency is incomplete documentation. It is critical to provide detailed information about the IVRT methods development, validation, and pivotal studies. Key details often missing includes aperture type and specification, dose amount, dose application techniques, ensure you fully coverage surface without bubbles, whether samples occlude or not, information about the dosage syringes, such as manufacturer, brand name, size, and believe sometimes if fixtures photo request. Explanation how to shear forces were minimized and control consistently across the different operators and between test and reference products. Precise study setup is important. It is important to stagger and synchronize those application and start times with the sampling times for each diffusion cells. This ensure precision in sample intervals is important. Deficiencies also ABC in method validation. Common deficiencies can be raised during the method validation. Parameters like membrane, receptor solution, sample solution preparation. There's important is when feedback is received, companies should conduct a thorough review of the point raised and respond with the detailed justification and correction. This involves ensuring the gap identified by the QA team or QC team are filled with clear, robust data discussion. Ensure high quality data submission with clear and complete information helps prevent deficiencies that could be classified as major, especially those related to the bioequivalence, I believe.
That's perfect. So I think for the time here that will conclude today's webinar. So Theo, once again, thank you so much for sharing your expertise. Big thank you to all the participants and the people who submitted questions. If we didn't address your questions, we will do it via email, as I've mentioned before. The webinar again will be available on the Teledyne Labs YouTube channel soon. So feel free to watch it on demand, share it with other people who may benefit from the content. For further information on this webinar or to access any other Teledyne Hansen content or products, please visit our website at teledynelabs.com or find us on social media. So once again, thank you, and we look forward to seeing you in our next webinar.
Thank you.